Hey everybody, I'm going to try and present to you a recording that was supposed to be for lecture 3 from last week. It sort of finishes up some of the animal material and then dives a little bit into the plant material with respect to this topic. So let me try and flip through this pretty quickly with you, and then we'll talk a little bit more about it on Tuesday. So we're on learning objective 2. So compare and contrast how lipid-soluble and water-soluble hormones act on target tissues. So you'll remember that we finished up starting to talk about neurotransmitters last time. So when we talk about chemical signals in animals, there's of course several different kinds of signals, but specifically when we're talking about hormones, these are chemicals that could be present in extremely low concentrations, but those concentrations are incredibly powerful. So they can have a really strong effect even in low concentrations. And they're going to bind specifically to hormone receptors. And we're going to talk about 3 different kinds of or classes of hormone receptors. They'll have specific receptors either on the membrane or inside the cells. And they're going to end up having major effects on whatever their target cells are. And we'll talk about the mechanism for how they're going to impact their target cells. And so it's slightly complicated, but I'm going to walk you through it. And these impacts can have a very long-lasting effect on the organism, and in this case, on the animal. Now, for you guys, you've all been through cells, so hopefully this is mostly a review for you, but just to recall some of the major features as we're thinking about form and function and how plants and animals can actually respond to these hormonal signals. So it's a pretty exquisite signaling system that allows us to respond to stimuli in the environment and also for us to maintain homeostasis in our bodies in the case of animals. Okay, so there's 3 chemical classes of hormones. So there's steroids, which most people are very familiar with. As we talk about these 3 different classes, there's 2 aspects of them that we're asking you to try to remember specifically. One is whether they're soluble in lipids or not. So lipid soluble, not lipid soluble, and where the receptors are. Are the receptors on the membrane of a cell or are they within the cell? Okay. In the case of steroids, as you can see here, they are indeed lipid soluble and they bind to receptors that are inside the target cell, which makes sense, right? Because these membranes are usually lipid bilayers. So if they're soluble, they can go right through the membrane, right into the cell, no problem. Do not worry. I am never going to have you recreate the chemical symbol for steroids. But if you still know that, that's great. The second class are peptides and polypeptides combined. So these are hormones that are not lipid soluble and thus they cannot pass through the membrane on their own. So the receptors for these membranes are always going to be, the receptors for these hormones are always going to be on the membrane. So they're then going to bind here. And remember, that means they then have to engage in signal transduction. So the signal from the outside is going to have to be transformed into an internal signal, which is then going to have some consequent actions. And we'll talk about that in a second. All right, so steroids, polypeptides and peptides. And the third class are the amino acid derivatives. Okay, so things like epinephrine. Okay. So these are, again, not lipid soluble, thus they have to bind to receptors on the membrane. So steroids, lipid soluble, will attach to receptors within the cell. Amino acid and the derivatives and peptides and polypeptides, not lipid soluble, so they're binding to a receptor on the membrane, and then signal transduction must happen. We'll get to those team questions later on in class. Okay, let's go through this in a little bit more detail. So with steroid hormones, right, here's a nice diagram that's basically showing you what's happening. So let's say you got a steroid hormone here. It's going to pass from the circulatory system towards the cell, towards its target. Now, it's lipid soluble, so it's going to pass through the membrane. The receptor for that hormone, for that steroid, is going to be in the cytoplasm. Once it binds, sorry, I'm forgetting to pull up my text. So once it binds to the receptor hormone complex, right, it's then going to enter into the nucleus. And in this case, what's going to happen is it's going to trigger gene transcription. You can see that right here, and that's in part because it's binding thanks to a zinc finger, which you should have done some reading on in your book and followed your reading guide along. And then once transcription has occurred, it's going to transcribe the mRNA, which is then going to be translated into proteins that alter cell activity, right? So it's going to come into the cell, bind to the receptor hormone complex, go into the nucleus, initiate transcription, which is then going to initiate translation, and then you're going to have an activated protein that's going to do something to alter cell activity. Now contrast that with what's happening with polypeptide hormones. So it starts out the same. Maybe it's born by the circulatory system. It's a water-soluble hormone, but remember, not lipid-soluble, so it cannot pass through the membrane. So it's going to attach, it's going to bind to a receptor on the cell membrane. In this particular case, it's going to cause the activation of a G protein. The activation of the G protein is then going to cause the activation of adenylyl cyclase, right? You can see that here. When that happens, that's going to cause the conversion of ATP to cyclic AMP, which in this case is the second messenger in the pathway. So remember, signal transduction, secondary messengers. The cyclic AMP is going to activate specific protein kinases. When that happens, you're then going to have protein kinases phosphorylating proteins in the cytoplasm. So that's what's happening here, right? And now you have these activated proteins, these phosphorylated proteins, and that's going to alter cell activity. So to remind you of some terms we talked about 2 lectures ago, so these steps 2 through 4, this is signal transduction. So this is transforming that external signal into an internal signal. You then have the production of a second messenger, and then you have this phosphorylation cascade, except I didn't really show you the steps in this phosphorylation cascade, but I'm going to show you that in just a minute. So just to remember, signal transduction is a stimulus outside a cell becomes converted into an intracellular signal required for cellular response. And a second messenger is a non-protein signaling molecule produced inside the cell in response to a stimulus at the cell surface. All right, so what about those phosphorylation cascades? Well, even this diagram doesn't go into the gory details, but think of it this way. Once that signaling molecule has bound to the receptor, you then have to have the activation of a molecule, which is then going to cause a series of chemical reactions to then, like a waterfall, fall into place. So I say here, protein kinases phosphorylate. So if you don't remember, adds a phosphate group to an enzyme, and it's going to activate them, and it's going to activate them in sequence. And so once this begins, it's going to take an inactive protein kinase in this case. It's going to activate it. That activated kinase is then going to activate another one, which is going to activate another one, which is then going to activate a protein, which is going to initiate a cellular response. Okay? So this is your phosphorylation cascade. And as you can see, it costs energy to do this. At each step, ATP must be burned in order for these protein kinases to get activated. We'll get to that in just next time, and we'll get to that next time too. Oh, whoops, sorry. So it turns out that you can have the very same chemical messenger impacting target cells in different ways. So here I say the same chemical messenger can trigger different responses in cells from different organs or in cells at different developmental stages, right? And this is because those cells are going to contain different receptors, different secondary messengers, different amplification steps. But maybe it's best to show you with a figure how this can be. So for instance, you can take epinephrine as the signal, okay? It's going to bind to either alpha receptors or beta receptors. And in this case, we're talking about the very same kind of organs. So we're talking about, the same kind of tissues, okay? So these are going to be smooth muscles, but maybe these smooth muscles are intestinal blood vessels, and this is skeletal muscle, okay? A skeletal muscle blood vessel. So when epinephrine, the very same molecule, hits the alpha receptor, that's going to cause this vessel to constrict. But when it hits a beta receptor, that's going to cause this vessel to dilate. So a very different response with the very same chemical, okay, with the very same molecule. What if we compare to having the same receptor, so same signaler, same receptor, but different tissues? Well, as we already said with epinephrine, when it bound to this beta receptor, it caused the vessel to dilate, but clearly it can't have the same response to a liver cell, right? Even though it has the very same receptor. So in this case, what will happen is it'll bind to the receptor. It's going to cause glycogen to break down into glucose, and it's going to be released from the cell. So you can have the very same chemical messenger that has a very different impact on different tissues and on different organs. And this is happening all over the body all the time. Okay, so I'm going to run through a couple of examples, of specific examples of how hormones affect animal systems. But in order to keep the video short, I'm not going to go over all of them. But when I upload the slides, you'll be able to see all of the examples within that slideshow. And then hopefully we'll touch on them a little bit on Tuesday. So we probably should have defined this earlier, but or discuss this sooner, but what do hormones do? So we're simplifying it here by saying that hormones are going to coordinate the activities of cells in 3 different big arenas. In development, growth, and reproduction, in response to environmental challenges, and in the maintenance of homeostasis. So we're going to go through each of these briefly. So if we start with animal development, and by the way, if you haven't taken a class in development, I highly recommend that you do if you're able, because it is a fascinating class that takes you from, the union of 2 cells to form that zygote all the way up to you as an adult. And it's a fascinating process. But to simplify it here, hormones play key roles in embryonic development, as you know, and there are a lot of embryonic stages. So you have hormones assisting in the growth throughout that process. So in particular, sex hormones play a key role in adults by regulating sperm production, the menstrual cycle, and reproductive behavior. So it's all about reproduction. So you're probably familiar with growth hormones. You may hear about these early on, particularly if you are involved in athletics and in developing in muscle development. You will have experienced this as you were growing. So as you grow, so you're fairly continuously growing from birth to reproductive maturity. But of course, everyone goes through these growth spurts during periods of their life. And that's usually very noticeable in that, clearly you grow very tall in a very short period of time. But you've probably noticed some side effects, like maybe your bones hurt. You have like mood swings during this, and or different parts of your body are going to develop differently. So those growth surges are stimulated by growth hormones, and these are produced in the pituitary gland. And this figure is meant to be highlighting some of the consequences of those growth hormones. So they regulate growth factors, which are signaling molecules that control the cell cycle. And so it normally will cause a decrease in adipose tissue and an increase in bone growth and in muscle mass. And so they're sort of highlighting this by showing a cartoon of a underdeveloped man and then a hyperdeveloped man. In the case of sex hormones, they have some similar yet different effects on both men and women. So generally speaking, you have muscle development in both men and women. You have hair development, but not necessarily in the same places. And frequently the skin gets oilier for everyone involved. And then, of course, you have secondary sexual characteristics that are involved and are influenced by sex hormones. So the development of genitals and breasts in both men and women are impacted by testosterone and estrogen. There's also the influence of thyroid hormones. I'm not going to cover this in more detail. You can read about this in the book and then take a look at this slide in more detail later. I would like to hit on this response to stress, however. When you're under stress, you have two consequences of this that are impacted uh, uh, by hormones. So what this diagram is meant to be showing you is here's your hypothalamus and then your pituitary in your brain, right? So if you're under stress in the short term, so let's say there's a fire that suddenly breaks out, or a bomb goes off, or zombies attack your house, that'll cause a nervous reaction that will then send a signal to your spinal cord and directly to your adrenal gland, causing the release of epinephrine and norepinephrine. And so these are responses that lead to short-term stress impacts. And so it'll cause your blood glucose level to go up. It'll cause your blood pressure to go up, so it's going to impact your heart. It's going to impact your diaphragm and your lungs. Your breathing rate's going to go up. It's going to cause your metabolic rate to go up, and you'll be hyper alert at this point. So this is what allows you to run and run fast away from a dangerous situation. However, if there is continued stress such that you're constantly responding in a short-term way, over a longer period of time, this can be quite bad for you, and it can cause an increase in your cortisol levels in your body, which ultimately will lead to the retention of water. It'll lead to increased blood volume and increased blood pressure and a suppression of your immune system, which are really bad impacts to stress. So hence, this is why you should do things like maybe study a little bit each day instead of like cramming right before an exam, which can be really stressful if you continue to do it repeatedly, right? A little bit of study each day can help you to avoid that stress. Just saying. I'll briefly go over this one too. Many of you, I'm sure, are very familiar with diabetes and the regulation of sugar levels in your bloodstream. This diagram is showing how your body can respond to blood glucose levels by regulating them, right? So if your glucose level goes too high, a signal will be sent to then your pancreas, which will secrete insulin. That will cause the synthesis of glycogen from glucose. So basically, it's taking that sugar out of your bloodstream and synthesizing glycogen from it, which is, of course, going to consequently cause the glucose levels to fall. And if they fall into the normal range, then the release of insulin will cease. If the glucose level goes too low, a signal will be sent to the pancreas to secrete glucagon, which then will, of course, break down glycogen into glucose. It'll catabolize glycogen, right? That'll cause your glucose levels to rise. And if you're at the normal levels then homeostasis will have been maintained, and you'll stop secreting glucagon from your pancreas. And so this is going on all the time in your body, this maintenance of sugar levels. I'm going to let you read digestion on your own in the slides and water and electrolyte homeostasis in the slides. But if you have any questions on these, we can definitely cover them in class or after class. We can meet as well. Same for blood oxygen homeostasis. So now let me try to dive into the plant aspect of this. So as I said earlier, most people seem to think that plants don't have hormones, but in fact they do, and they have some really interesting impacts to plant systems, but you can imagine that they work differently from animal systems. So let's go through a few examples. So there's two things to note about plant growth regulators or plant hormones. So a single hormone may affect many different target tissues, carrying a similar message to a variety of tissues and organs. And several different hormones may affect the same response as they interact with one another. And so let's take a look at some examples. So crosstalk is the interaction between signaling pathways triggered by different hormones, integrating information from many sensory cells and signals. So there's 5 different hormones that we're going to talk about, and we're actually going to come back to these at a few different points in the rest of the class, and you're also going to be talking about some of these in lab. So gibberellins, auxins, cytokinins, ethylene, and abscisic acid. So gibberellins are produced in shoot tips They can also be produced in immature seeds and in anthers. So that's the part of the flower that's producing pollen. So the impact of gibberellins is to promote stem growth, and it does it via cell division and cell elongation. Okay, but it's not the only one involved in both cell division and in cell elongation. It promotes seed germination, and you can see that happening down here in this picture. Auxins are also involved in cell elongation. So also produced in the tips, in the shoot tips and in young leaves. It's involved in phototropism and gravitropism. So when it comes to shoots, when for those that move towards the sun or that track the sun, right, it's auxin that's involved in this. And here's how it happens. So you have auxins that could be found throughout the cells of a young shoot. When the sun moves in a certain direction, on the shady side of that plant, auxins begin to be broken down, but they're maintained on the sunny side, they're broken down. They're maintained on the shady side. What happens then is that when they get into the cells on the shady side, they cause these cells to elongate. And when these cells elongate while these remain the same size, they cause the tip to bend and face towards the sun. So that's how plants are able to actually move and track the sun. So auxins can also promote differentiation of xylem and phloem cells. Cytokinins are synthesized in root tips. They're going to promote cell division, and so they can be used not only to promote root growth, but they can actually be transported throughout the plant. And then they can go further up into the plant and promote chloroplast development. And so with cytokinins, you get more mitosis, which means you get more cells, which means you get plant growth. Ethylene is one that you may already be familiar with. So this is a chemical that often gets sprayed onto fruits when they're picked. So when they're first picked from their groves, whatever the plant is, often they're picked when they're green and they're not yet ripe. And so ethylene will be applied to them to help promote the ripening of those fruits prior to when they normally would be ripe. It is a chemical that's produced in all organs when the plants are under stress. And it's strongly associated with three aspects of senescence or death. So fruit ripening, which you see here in these images, flower fading, which is a form of death, right? And then leaf abscission. So this is when the cells within the junction between, say, the stem of a leaf and where it's attached, they die, and that causes the leaf to fall off the tree. And so you're seeing leaf abscission all around you right now. Leaves are falling off the trees like crazy. But that's not necessarily due to just ethylene. It's probably also due to abscisic acid, and more so right now, abscisic acid. So this is a hormone that's produced in almost all cells within a plant. They can have the impact of inhibiting bud growth and seed germination, and they induce closure of stoma in response to water stress. So this makes sense, right? So at a particular time of the year when it's maybe time, in decreasing light cycles and maybe decreasing temperature, right? That's probably not the best time for a plant to be growing or germinating seeds. If there's still seeds on the ground, it's definitely not a good time for buds to be growing. And so you probably do want to close your stoma in response to water stress. So abscisic acid, so at about the same time that leaves are falling off the trees, these same processes are going on as well in preparation for winter or for drought or conditions like that are not conducive to plant growth. All right, that's all I got for now. Take a look at your reading guide and review it. And then any questions that you have, we can cover those in class on Tuesday.